can start now. Alright. So we have the small arteriole. We call it arteriole. It's not an artery, it's arteriole, and that's coming inside the nephron. So we call it afferent arteriole. And this arteriole, it divides into smaller capillaries, lots of smaller capillaries. And these smaller capillaries have a fancy name. We call them the glomerular capillary. And then these capillaries, they join back and they form another arteriole. And this arteriole is called the efferent arteriole. So for example purposes, you need to know the names. So this is afferent arteriole, then the ones coming out is the efferent arteriole, and the capillary that has sort of split into smaller blood vessels, we call them glomerular capillaries. In the book you can call this area glomerulus area. So this is the glomerular capillary. So what you want to do is that you want to filter all the stuff out. Okay, so I'm going to cross this out. And obviously you have lots of capillary here, glomerular capillary. I'm going to draw in one capillary as an example. So this is one of the capillary, small capillary, and in there I have lots of water. So you want to take water out of your body. Okay, you also excess water. Okay, you have like mineral ion, like sodium. You also have like glucose, G4 glucose. You also have larger molecules like red blood cells, white blood cells. You have proteins, albumin, and so on. So what happens is, all of this needs to be filtered out. Now, it's going to be filtered out into a capsule. Now this capsule here is called Bowman's capsule. But AQA also allows you to call it renal capsule or renal capsule. Both words are fine. This is a capsule. This is where I'm going to take the filter into here. So the stuff that's going to come out, we call it filtrate. But it's coming out from the glomerulus. So we call it the glomerular filtrate. So I'm going to basically just draw it in a simple way. So all of these needs to be filtered out. So basically it just comes out from the capillary. And as you know, capillary, they're very thin membrane. It goes through the membrane. So water is very small. Sodium is very small. It does not need a channel protein. It just diffuses through the phospholipid bilayer. So it's sort of diffused through. And between these capillary, they are made from cells. These cells are called endothelial cells. Not epithelial, endothelial cells. So endothelial cells, between the cells you have like small gaps, and that allows maybe like glucose to get through and so on. So things are filtered out, okay, and then it goes into here. This is where you store it, and this is the capsule, renal capsule. Between here and here there are 3 layers, and that's quite confusing. That will be in a different video. Now how does it leave? How does it get filtered from the capillary into here? The afferent arteriole, the diameter is larger. So its diameter is larger. The efferent, the diameter is smaller. That creates a pressure here. You have to know the name of the pressure. Increased hydrostatic pressure. Hydrostatic pressure. And that pressure allows you to filter everything. That's why it's called ultrafiltration. So you can filter everything out except big things. So you know that between the endothelial cells of the capillary, very small spaces. So large things like blood cells can't get through or should not get through. Protein should never get through. Okay? Smaller thing will get through, like water, mineral ions, urea is a waste product, urea, and also glucose. Although glucose is not that big, it's quite small. It's C6H12O6. It's not that big. Can get through. That will get through, except the big stuff.